Hello and welcome to BioCode. In this video, we will see how we can utilize sequence records or Seqrecord library from BioPython, which is basically a module of functions that provide us the ability to create sequence records and that resonate with the sequence records of GenBank, EMBL, EBI, and other databases. So, for example, you must be familiar with FASTA sequence by now, and you would understand that each FASTA sequence has a sequence, an ID, a description, that sort of stuff. So, in this video, you will learn how to actually create sequence records that resonate with that scenario. But first, you need to understand the look or the data type, which basically we are trying to imitate in Python. So, basically, a sequence record is a module within BioPython that allows you to create sequence records for various reasons. Let's suppose that you are working with a huge amount of list of sequences and they are just random sequences, and then you would like to designate ID, designate quality, designate annotation, designate description to each of their sequence. And to do that, obviously you can't do that manually because it is going to take a huge lot of time. So obviously you are going to do it through some programming languages, and Python is the way to go for that. And let's suppose that you're working on 100 sequences and they are all sequences, they are just strings like this, and let's suppose this is a protein, let's suppose. So they are like this and you are working on all of them, but obviously each of these or each of that sequence represents something in your research. Each of that sequence should have some sort of ID, should have some sort of description, should have some sort of annotation features, or some sort of any biological annotation. So to do that you need Python language, but to do that particularly you need sequence record module within BioPython. So first of all, I'm going to demonstrate that what actually is FASTA file if you do not understand it by now, but I'm going to explain it more. You need to watch for our FASTA versus GenBank video and FASTA format and GenBank formats video for to understand this particular video and the further videos that are down the road. But otherwise, it will not make sense to you. So first of all, this is a chicken sequence, sequence of a particular gene, and the name is given over here. It belongs to Gallus gallus, which is a chicken. So any sort of sequence, this is just to provide you an example that which species it belongs, but to any sequence, whenever you are retrieving a sequence, it should have always an ID like this, a description like this, and a sequence like this. So this is a bare bone FASTA structure, unless it is a FASTQ file. But this is a FASTA structure, and it will always have an ID, it will always have a description, it will always have a sequence. So in this video, we are going to imitate that sort of scenario, that we are going to create the exact structure like we see over here on this page. This is a GenBank page on FASTA, on NCBI, but it is a FASTA record. So sequence record module can allow you to create GenBank records, can allow you to create FASTA records. That is not the problem, but in this video we will see how we can do that. So let's suppose, obviously, according to this video, according to this particular page, we need a sequence ID, a description, and a sequence, right? So let's suppose that we have a sequence. Let's just copy it from this page. Let's just copy this particular first line. Let's consider that this is a entire gene of the Gallus gallus chicken, and we have created a sequence object. If you do not know how to do that, you need to watch the previous videos. We have created the sequence object. Then we are, as you can see, we have imported sequence record from the BioSeq module, and in that sequence record function we are passing the sequence object that we created over here, and it has now become a sequence record. So you must be thinking that will this particular sequence resemble the sequence over here? No, it will not, because we have not provided any ID. It will however represent some sort of representation of a sequence record, but because of the fact that it contains a sequence, and but it does not contain any ID, does not contain any other information. So let's just run this code, as you can see, I'm printing the sequence record, I'm printing the type of the sequence record variable. Let's rerun this and we will see our results. So as you can see, id is not unknown, we do not have any id, we do not have any name, we do not have any description, number of features are unpresent, but sequence is present. As you can see over here, the sequence is present. And in the next line we are actually able to see that it belongs to a class of sequence record. So that was fairly easy. So let's go back to our original code and let's go back to the main file. So now what I'm going to do, I'm going to assign an ID. I'm going to assign an ID over here. And because of the fact that it's unknown, so I am assigning an ID that is random abc123.1. And then I'm also providing a description. How to do that? Basically, you need to understand how to do that. Sequence record provides you various information that can be utilized to assign or retrieve various information from that particular sequence object. So you must be thinking how to understand what sort of information is available for us. Let's suppose that you have a sequence record. It will have 5 or 6 properties depending upon what sort of sequence record it is, but it should always have .seq property. Like over here, it should always have .seq property in just like this one .id. It should always have .seq, but if you just enter it over here, and let me rerun this code, and you will then see that it is indeed a property. Okay, so here we go. You can see that it is a representation of a sequence directly taken from that. So let's go back to our original document, and so basically it should always have .seq property. It will always have .id, and you can see that we do have .id, we do have .seq. Sequence object always has .seq that actually basically retrieves or assigns a sequence to that particular biological object that we created in Python. Then it will always have an ID. ID will always be alphanumeric, or just to represent GenBank. You can create your own. You can. It could be from the file you are parsing, you are reading in Python. Then it will always have a name. A name will decide that it can be, for example, if you go back to our original output of the video, you can see that there is a property as name, but it is unknown name. We did not define any name, so we can do that as well. We will learn over here. Then we have a description, and you can see that we do have description over here. Then we do have letter annotations, which basically will hold the annotations. Then we will be able to apply annotations to our objects. Basically, FASTA files do not have any other information such as annotations, letter features, or that sort of stuff, because it is bare bone structure and not GenBank. Whereas on the other hand, GenBanks do have dbxrefs features and that sort of stuff. So you need to understand those. So these are all the available properties of these sequence record objects, and that you can manipulate to retrieve or assign values. So how do we actually retrieve values? If you need to retrieve value, just utilize the code as it is. It will allow you to retrieve the code. But when you need to actually assign the value, you need to assign it using the equal operator, and you will be able to assign the values. So first of all, we have learned that we can print out the sequence object. We can print its type. Let's remove it because we have learned that. Now I'm assigning an ID to this sequence record. Then I'm assigning a description. Let's also assign a name as well. I'm going to make it name, and in that name